Rob-See-Co Future Farm. Today's innovations, tomorrow's solutions.
Hello and welcome back to the Rob-See-Co Future Farm. I'm your host, Jim Robinson, and here we are at Future Farm overlooking our soybean field, and we are going to talk about biostimulants, biofungicides, all sorts of biology. So I'm here with Allison Jacks of new Leaf Sumbiotics, and Allison, welcome.
Thank you. Thanks so much for having us out.
So Allison, you guys have been working with us to help supply a set of our biological control, biostimulant types of products, and we got quite a few different trials out this year. And so can you tell me a little bit about what types of products are we trialing in our trials this year?
Yeah, I've had a great season kind of tracking these trials, right? So we have our cotton biostimulant and our TS601 EPA registered biofungicide together in a cotton field outside of Houston. That one's been very interesting to scout. And then we have our corn biostimulant and TS201 EPA registered bioinsecticide on silage corn outside of Columbus in Ohio. And then also the soybean trial here where we're looking at our soybean biostimulant and again that TS601 biofungicide. So excited to see how these are developing throughout the season.
Excellent. Now before we take a look at these samples, which I'm really excited to show, tell me a little bit about, you say these numbers, TS601, TS201, so biofungicide, biostimulant. What can we expect from a TS601 type of a product? What will it do?
So what's unique about New Leaf's biocontrol technology is that the microbes that we work with are PPFMs, or pink pigmented facultative methylatrophs. That's why everyone calls them PPFMs for short. They are able to help the plant better defend itself. So these aren't a direct mode of action, they're an indirect mode of action. It almost acts like a vaccine where it can turn on different genes in the plant and help it be poised and ready to deal with biotic challenges once they come, right? So it's basically stimulating the plant's own defense systems and providing the benefit in that way.
Excellent. Now, these are all different types of biostimulants, biofungicides that you don't necessarily apply to the leaf of the plant, right? How does it get onto the plant itself?
Yeah, so these are all seed applied, and they're seed applied as a dry planter box treatment. And because of this unique mode of action, one, they colonize the plant really well. So if they're there as the seed germinates, they grow with the plant. They colonize the roots, colonize shoots and leaves, stay with the plant for the entire season. And in that way, you don't have to reapply, right? They're natural plant colonizers. One of the fun things about them is their pink color. That's actually a sunscreen. So that's a carotenoid pigment that allows them to live very well on the leaf surface without drying out or getting sunburnt, basically, right?
Oh, that's fantastic. Yeah, so let's get to the samples here. You guys did a little bit of scouting here as you got in the other day, and so tell me a little bit about where did these samples come from? What are we looking at?
So you had, or one of your partners had a side-by-side trial in northeastern Kansas. So these were soybean, I believe, planted into a cover crop and corn stubble, so like no-till kind of environment. Beautiful soils, very like dark, rich soils, a little bit hard to dig, but we made it, a little bit hard to drive into after a rain. But so there were 16 rows of New Leaf's soybean biostimulant, and then TS601 biofungicide, and then 16 rows of grower standard practice.
Excellent.
And what we also like to see is how people use these in a system, right? So none of these are standalone biocontrol, but they're a very important tool in an IPM toolbox, right? So can you improve upon grower standard practice by adding in another mode of action?
Absolutely. And so, when we laid these out on the tailgate of the pickup, it was immediately apparent that there are differences between the grower standard practice and the treated TS601, TS201 treatment group. And so, if we look just at these plants, there's clearly a difference in stem diameter. That was the first thing that was really apparent to us, was the stem diameter.
Absolutely, yeah.
You can also see a significant elongation or size difference in the taproot of these plants. And then, you know, we were looking through, and we haven't done the counts right off, but you can pretty clearly identify that there are more blooms on the treated versus the untreated, which, as we talked about before, soybeans are a very plastic dynamic plant.
Yeah.
So it doesn't always result in yield, but boy, does it set you up in a good spot for yield. Now, what do you guys typically see with these types of treatments?
Yeah, so we've tracked a lot of the root phenotypes, or just like what they look like and how they develop. We see a lot of early vigor enhancement, and we've got some great data sets where we've imaged thousands of root systems, analyzed them, and are able to show, yes, there are more fine roots, there are more root tips, there's deeper soil penetration. That's a really consistent phenotype that we see with our soybean biostimulant and also our corn and cotton biostimulants. Again, those are all different species of microbes that are like tailored for that crop, but once we find the right microbe crop combination, we see some of the same general effects on the plants.
Exactly. And you know, if I'm a farmer, what I'm thinking about is, okay, you know, that's all well and good. There's, you know, better taproot, there's more stem diameter, more blooms, more plant size, vigor. But what does that mean for me? I mean, you know, if you could just walk us through, why are these factors important in establishing yield?
Well, I mean, what we see is, like you said, you set everything up for success, right? The rest of the season obviously has to line up in terms of other things happening to get all the way to yield. But with our biostimulants, we're seeing over hundreds of on-farm trials, 70 or higher % win rates, right? And that's really phenomenal for a biological where we see really consistent responses. So that early vigor. We also see enhanced nutrient uptake. So with our biostimulants, they're able to help the plant capture more micronutrients and basically convert them to a plant available form. So especially with soybean, we have interesting trials going back a couple years showing a real benefit with IDC if you have those high pH soils. So yeah, our technology can make micronutrients more available. So with that increased nutrient uptake, the rooting enhancement, we typically see a pretty consistent yield response.
Yeah.
Even in high yielding environments.
That's exactly right. You know, I always like to think of it as farmers harvest grain. The plant is harvesting sunlight and harvesting nutrients from the soil. And so by building a larger factory for that plant to harvest nutrients and sunlight, you can harvest more grain at the end of the year.
Absolutely.
So Allison, these structural plant differences that we see here, the larger stem diameter, the deeper taproot, these are from the biostimulant, right? What would we expect to see out of the biofungicide as we get into a more of a disease typical year?
Yeah, so once a pathogen kind of kicks off within a field, what we've been seeing with all of our years of development of this is delayed incidence. So even in inoculated trials where the researcher put the pathogen there, it maybe takes a couple more weeks before the symptoms are even visible. So you get that delayed incidence, and then you get about a 50 to 60% decrease in severity.
Okay.
So again, that's a great tool to have in rotation with other types of tools in terms of disease management. And while no disease has really raised its head here, I looked, right? We scouted, but we saw maybe some thrips, but really no fungal or bacterial pathogens. What we've seen in your trial in Texas, we saw rhizoctonia early on. You could very clearly see the lesions on the stems. And then when we went back to visit later, we saw so many more cotton plants having had survived that rhizoctonia that it was harder to walk through the field in the treated areas. And it was great because the grower was able to say, wow, you know, we struggle with these early seedling rots. We do use, of course, the on-seed treatments, but this rhizoctonia was breaking through that. And having the 601 on top of the base F&I on the seed allowed so many more seeds to survive that we could tell as we were walking through the field. So yeah, and we're excited to see that one, you know, all the way to the end. Yeah.
Yeah, that higher population means a lot for the grower. It can help improve yield. It can do a lot of things.
Yeah, exactly. So again, while we didn't see any disease in this field yet, we're excited to come back, see if there is maybe, white mold or SDS that shows up later in the season to really track that effect of the biofungicide.
Oh, it'll be fun to come back in 5 or 6 weeks to see how that turns out.
Absolutely, I'm looking forward to it.
Thank you so much, Allison, for joining us. This has been a lot of fun talking this through.
Thanks so much for having me.